Hello friends, welcome back to my channel, and today in this video we are going to study about gene mapping with recombination frequencies. Morgan explained that physical distance between genes on a chromosome are related to the rates of recombination. That means that the distance between the genes, suppose two genes are present A and B. So the distance between A and B are related to the rate of recombination. We already learned that if two genes are very closely packed, the rate of recombination is very less. That's why we called such genes as linked genes. So that means physical distance between the gene play an important role determining the rate of recombination. So therefore, recombination frequency could provide a convenient way to determine the order of genes along a chromosome and would give estimates of the relative distance between the genes, because we are saying that distance between the two genes are related to the rate of recombination. Therefore, if we know the rate of recombination, then we can easily determine the distance between the two genes because these are connected, the distance between genes and the rate of recombination. If we know the distance between genes, we can tell the rate of recombination. If we know the rate of recombination, then we can say what is the physical distance between both the genes. Now, what do we mean by genetic maps? Genetic map, it is a chromosome maps which is calculated using recombination frequencies. On the basis of recombination frequency, when we map the position of different genes, we call it as a genetic map. In contrast, what do we mean by physical map? Physical map is the map that based on physical distance between the genes. There is a difference between genetic distance and physical distance. Genetic distance means the distance based on recombination. Physical distance means the actual distance between the two genes. Map unit, it is the measurement of distance on a genetic map denoted by MU. One map unit means 1% recombination. Map unit are also called centimorgans after the name of the scientist Morgan, and 1 morgan is equals to 100 map unit. Now we will see how we can use the recombination frequency to map the genes. For this, the first step is to find out the gene pair having the maximum recombination frequency. So here the gene pair is C and D with recombination frequency of 23%. That means in a chromosome, C and D genes are the farthest gene pair. 1% is one map unit. So 23% of recombination frequency means 23 map unit. Now the distance between B and D is 13%. So from D, approximately here, we can say the B is present with the distance of 13 map unit. Now the distance between A and D is 18%. So from D, again A can be here with a distance of 8 map unit. So like this, on the basis of recombination frequency, we can map the gene position. So in the final chromosome, what we can say, the gene position is C, then B, then A, and then D with the different map unit given here. So this how we can determine the gene position. Now there are few points which you have to keep in mind for determining the gene position. First of all, the recombination frequency between two genes cannot exceed 50%. This we are already learned that recombination cannot be more than 50%. Now 50% rate of recombination can also be for the genes located on different chromosomes. If we are saying that the distance between A and B is 50 map unit, or they have the recombination frequency of 50%, that means maybe they are present on different chromosomes. A is present on some chromosome number 1 and B is present on some chromosome number 2. Or even if they are present on single chromosome, they are much far away from each other. So 50% is the maximum limit. If two gene pair is present on single chromosome, it has to be less than 50% recombination. If the recombination is given that if the two gene pair is having 50% of recombination frequency, we can put them in different linkage groups or in different chromosomes. In one linkage group, two genes cannot be equal to or more than of 50% of recombination frequency. Now it is difficult to distinguish between the progeny produced by double crossover and the progeny produced when there is no crossing over. How? This is one chromosome, this is second homologous chromosome. When there is a recombination between capital A small a and capital B small b. If it is a single crossover, how we can expect? This is A, this is B. This is A and this is B. In case of single crossover, if the crossing over is taking place here, then we will get the sequence like so this is single crossover. But what is happening in double crossover? As we can see here in the center. So two times the crossing over is taking place between two genes A and B. So because of which what happened, the central blue and the red part got interchanged. If I have to show you here, this is our homologous chromosome. The crossing over is happening two times between gene A and B. So now how the gene will behave? This red section, then the lower blue, again the red section. Why? Because from this red, then blue, then again this red. Clear? So here the gene is like A and here B. So when there is no crossing over, then also it is A, capital N, capital B. After double crossing over also it is capital A and capital B. So if we are seeing with respect to capital A and capital B, then we will say that no crossing over has taken place because we don't know that the crossing over has taken place twice in between and the exchange has happened because we are only mapping with respect to the genes A and B. Therefore, it is difficult to say that crossing over has happened if there is double crossovers instead of single crossover. So in case of double crossover, it is difficult for us to say that whether crossing over happened or not. So these are few points that you have to remember. The most importantly, 50%. This is your maximum criteria. If the two gene pair is given recombination frequency 50%, then definitely maybe they are present on different chromosomes, or if they are present on same chromosome, then they are present on different linkage groups. Now this is a homework for you. Some gene pairs are given and recombination frequencies are given. You have to map the gene on the basis of these recombination frequency.